So this lecture will be about the genotype and phenotype difference and the different ways in which alleles can interact. So let's take a look here at the little drawing that we have. So the drawing I'm looking at right now is this drawing right here. And I'm giving the example in this case right now of the thumb. And I use the hitchhiker's thumb. And I know this doesn't look like hitchhiker's thumb, but that's what this is supposed to be over here. It's supposed to be a bent thumb. It looks ridiculous, you know, like that. And then this is a straight thumb when you sort of stick up your thumb and it's straight. And I guess I could put this on the rest of a hand, right? Oh, that's even uglier. Okay, well, in any case, you get the idea. It's a simple trait that isn't really good or bad. It's just different. And it behaves like a trait that is driven by a single allele. Now, if you really get interested in these things and look them up, you'll find out that some of these traits are actually driven by multiple different genes. So it may be a little more complicated than I'm laying it out, but it turns out that most populations, about 95% of the time, inherit these alleles in such a way that it's pretty much straight up what we call a simple dominant gene or allele interaction. So what I did was I give you this idea over here that my two copies, your two different homologs, right, of this particular chromosome, and I put the thumb gene is on here in this place, and then you can see that I've got the straight on one and curved on the other. So this individual has the gene located for thumb shape is the gene, right? That's what we call the gene, and this is the curved allele, and we have the straight allele. And I've labeled them over here. We can look at T sub 1, I'm calling, for thumb shape with the curved allele, and T sub 2 with the straight allele. So, of course, if an individual has both alleles, because they're heterozygous for this particular trait, they can't show both curved and straight. It's not like your right hand is curved and your left hand is straight. These two alleles interact in some way and generate your phenotype. All right, and so that interaction is what I want to talk about today. And let's look here. I have genotype is the actual allele combination. So that's T2 and T1. That's the actual genotype of a heterozygous individual. But the phenotype is the physical appearance or expression of the genotype. So that's how an individual looks. It depends on the allele interaction. Sometimes the phenotype is actually directly tells you the genotype because it's a very clear, there's no interaction, like both alleles are the same. Let's say that, right? It's a homozygous individual. On the other hand, if you have heterozygous individuals, sometimes the allele interaction doesn't, the phenotype does not tell you everything about the genotype. So this depends on allele interaction here. So one kind of interaction is called the simple dominant one. All right, and what that means is in simple dominant case, an individual can have a T1, T2 genotype, and yet they have the straight thumb. And what this means is that T2 is expressing over T1. So what we use is this cool word mask down here. Yes, masking. So which means to cover the other allele's expression. And this is a word that saves you time in trying to explain it. T2 masks T1 in the phenotype. That's easy, right? So you could just write T2 masks T1 in phenotype. So now you know if an individual has a genotype of T1, T2, that they will actually have a phenotype of T2. That means T2 is dominant. Now, it's really important not to confuse this with the idea that T2 is stronger in some way or more likely to be inherited in some way. It is more likely to be expressed when it's there with the T1, right? So you don't see T1, but that doesn't mean that you go back to metaphase in meiosis, that it's any more likely to be handed off to the gametes, and it's not. So this is all about which one is more likely to express, not which one is more likely to be inherited, because they're both equally likely to be inherited. Now, one possible explanation that I was trying to come up for this hitchhiker's thumb is that maybe the allele actually, like this is one way of thinking of these, there might be a mutation in the hitchhiker's thumb here that such that a piece of the joint is missing right here so that it doesn't stop the thumb from kicking back like that. And it's a small mutation, a change in the genome changes the proteins here, and that results in this new shape, okay? And so now that also would help you understand why having one T2 allele, right, would be sufficient to make an okay shape here, and so you wouldn't have the bent thumb. That's not necessarily exactly how the thumb works. I'm just trying to give you an idea for how ways in which genes do work in some cases. So this is one possible explanation, right? So if an individual has a genotype of T1, T1, then they're going to express T1, so they'll have a curved thumb. This is called the homozygous recessive. And this one over here is homozygous for T2, which will show the T2 straight allele. Okay, so we have the different names for these, and I probably should mention them, because when the two are the same, I can put it over here. When you have T1, T1, you have homozygous. T1, T2 is called heterozygous. And T2, T2 is equal to homozygous again. Now, in the case that I have, this T2 is the homozygous dominant case. So you can call this homozygous dominant, heterozygous, and homozygous. And this, we would call this other one the recessive case. Recessive alleles are not less likely to be inherited. They just are less likely to show up in the presence of the dominant allele. So they only show when there's two of the same kind. All right, so let's look at some of the other interactions. I'd just like you to know what these are. So, I could ask you like what sort of phenotypes might be the case for codominant, for incomplete dominance. So I've got some different categories. In a codominant case, we're mostly looking at this heterozygote, right? This individual here that's T1 and T2. In this case, when they're codominant instead of simple dominant, then the heterozygote forms a third phenotype. So what that means is you might have a white phenotype for T1, T1, and a red phenotype for T2, T2. Say this is flower color. And then you would make a sort of off-the-wall third type, which would be the yellow color, which has nothing to do with the other two. Blood type is like this because you're type A or B, or you're type AB, which is a third type. It's not really A or B. It's not even intermediate to them because it's both. Another possible interaction is incomplete dominance, and that means that in the heterozygote, the phenotype is intermediate to the two homozygotes. So that's a white, red, pink flower. The pink is pretty clearly intermediate to red and white, right? That would be called incomplete dominance. In both of those, of course, you can see the genotype in the phenotype. Now, the other type that I like to talk about a little bit is quantitative traits or multiple alleles in genes. And what this means is that the more you have of something, the more that you produce of it. And if you have many different genes, each with two alleles, you can have a lot of alleles that produce that. So my example here is an example for skin color, because both hair color works this way and skin color works this way. And take two genes, A1 and A2. A1 produces melanin, which is a UV protectant. And then A2 would be a gene that does not produce melanin, so you don't have the UV protectant. It also produces a dark color. So when we look at this, we might have we're going to look a little bit at the biology of that. When you get to skin lab, you'll see that this is like a skin layer. And when you look at hair color, you'd see there are melanin producing cells down here at the bottom of the tissue that produces the hairs, right? And that's what does the coloring. So if you have melanin producing cells, 2 of them produce lots of melanin. If you have only 1, you produce half as much melanin. If you don't have any, you don't produce any melanin, and you get blonde hair. So you can basically have really dark hair, you could have really light brown hair or blonde hair, depending on if you have none, 1, or 2 of these melanin-producing cells. Now, we can also imagine how this might carry across multiple different locations in the genome. So say you had 10 sites. I think I did this with 5 sites in class, where you had actually 5 locations on each of your chromosomes where you could have these alleles. And so what that means is that you have an opportunity to have as many as 2, 4, 6, 8, 10, 12 on this example. 2, 4, 6, 8, 10, 12, 14, 16, 18. You could have up to 20 melanin producing cells. And so if that is the case, then each one is either melanin producing or melanin not producing. So you could have up to 20 different alleles, which would produce very, very dark hair. Or if you had absolutely none out of 20, then you would have very, very blonde hair, right? So this is how the phenotypes actually sort out sometimes, is that some people have half and half, right? And that can be 2 at one, none at the other, some at one, heterozygous. You could, whatever the mix, you count up all the alleles, and you might get a distribution like this of hair color, such that some people are really blonde in hair color, and then you start seeing them get darker, and some people have super, super dark hair colors. So as a result of this, we tend to get a normal distribution of traits, and we find that, and we call this a quantitative trait because the more of an allele you have, the more of the phenotype that you have, right? So that's what quantitative refers to. So, we can also apply this to, somebody asked me in class about red hair, and some of this is on here because of the red hair. I didn't know what exactly causes red hair, but I looked this up for you guys, and it says melanocortin-1 receptor is also called the melanocyte stimulating hormone receptor. And that gene, when that gene gets mutations in it, it can cause red hair. And it gets a little more complicated after that, compound called pheomelanin instead of melanin. And so there's different forms of the melanin. And so as a result, you can get red hair. It turns out, though, that there's like 8 different genes involved in red hair. So I like to say it's not as simple as blaming the mailman if you're the kid in the family with red hair. It turns out that it is a little more complicated, and that there are multiple ways to get red hair. And there are multiple kinds of red. So that's kind of fun. Probably the most common reason why children show up with red hair with parents that don't have it is that if you have the dark colored alleles, then even if you have red hair alleles, you can't see the red. So that means that in order to see red, you have to have no dark alleles. So you have to be blonde, and then you can see red if you have it. So it's a little bit tricky that some people actually do have red in their hair, but you can't see it because their hair is really dark. So they could hand off the red gene and not the dark hair genes. Some of these genes also work like this for height. So there are height alleles or height genes, many of them, that contribute to things like bone elongation rate and maximum rate and all these different things. And so if you get more tall alleles at the gene sites, then you end up being taller. And if you get fewer, you end up being shorter. So, whoa, how about that, huh? Let's see, what else do I have in here for you today? Dark skin. Somebody in class asked about skin, and this is also produced in a similar way. It's also melanin, and it's melanin producing sites. So I drew an idealized picture here where this individual has 1, 2, 3. You can count up how many non-melanin producing alleles they have, and this is another person that actually has on this side would actually have more melanin producing alleles. And so when they inherit them, there's a possibility you could hand off all of these, but you could also hand off all of these, right? So it's a little bit tricky to know. There's a lot of chance in the game, but it is true. Well, let me tell you quickly how melanin works. This is your skin cells, right, which are these sort of squamous cells, which means they're square, right, and they're on your skin. And deep down near the basal layer there are these melanocytes. And these melanocytes down here produce the melanin that sort of spreads around in the skin. And everybody pretty much has the same number of melanocytes, but they're upregulated in some people. So with the more alleles you have for upregulation of melanin, the more melanin you produce. And so it isn't because that people with darker skin have more melanocytes, because they don't. Their melanocytes just produce more melanin. So, and that's what actually causes the skin color to change, right? And it is very adaptive in sunny climates. And let's go down here to take a look at heterozygote offspring. So this was an interesting cross I was making. So when two people have children, right, and you take a very dark-skinned person and they make a child with a very white-skinned person, what you can find is that if they have, let's say that it makes it easy for us, we'll say they have 10 children, all those children are going to be heterozygotes, because each of them, like if all you have is A1 in your genome, that's all you're going to donate to your offspring, and all you have is A2, that's all you're going to donate. So when you make sperm and egg, you make them all with one kind, so everybody turns out to be heterozygous. So they're all sort of, they're like a tan medium color, right? But then when those heterozygote offspring take up mates, right? They have children, and then there's a tremendous possibility for them to have different combinations. So each of them has got 50-50 in sort of a mixed up way. And in meiosis, that means that this person over here, well, either one of them could potentially give all of their sort of melanin producing alleles to the offspring, or they might produce none of them for the offspring. It's quite possible that it could happen because they're all heterozygotes at each site, right? So it's also possible they'll give off an intermediate number. So if they have 10 children, then you get children that are all in the same family, and some of them have very light skin, and some have very dark skin, and some are in the middle. So that is the greatest diversity of potential offspring phenotypes, which is kind of interesting. Great. So I'm going to end this lecture here. Quick review. The main ideas here were the interactions of alleles being the main ways, like we went from genotype and phenotype and looked at the alleles, and then we talked about simple dominance, codominance, incomplete dominance, and quantitative traits. Thanks.